Good afternoon. My name is Tom Randolph, and this presentation is a brief overview discussing tropical tree dendrochronology research. So, dendrochronology, as you know, has a long history of illuminating a variety of areas of scientific discovery: environmental science, archaeology, historical research, climatological research. They've all benefited from this innovative discipline. There are, though, areas where research in dendrochronology is really challenged, and one specific challenge involves identifying reliable annual tree ring formations in tree specimens growing in tropical latitudes. One example would be this guava berry photo that I took here on the left. It can show you some of the challenges that these rings can produce. So this presentation is going to be looking at highlights from some of the interdisciplinary approaches used to improve signal-to-noise ratio of tropical trees, even when they do produce idiosyncratic growth ring characteristics. Here on the left, you see a baobab tree growing in front of the ruins of a sugar plantation here on St. Croix. Tropical trees in these latitudes don't give clear ring growth signals. Quite often, tree growth characteristics in tropical latitudes are dependent on precipitation rates, temperature-driven evapotranspiration, root depth, and even elevation can begin to give signals that would normally in higher latitudes be associated with annual growth and annual growth rings. Ramirez et al. Explains that one of the key challenges with tropical trees is they do not produce consistent annual rings that reliably record the annual growth of a given tree. Furthermore, climate-related signals are difficult to validate because of the difficulties related to noise, dating calibration, and cross-dating of tropical tree samples. So these trees do provide some very difficult challenges for dendrochronology. On the left, you see a sample, a cross-section of the guava berry tree. I was able to obtain this locally and acquire some sandpaper, and I was able to sand this down a bit to look at the rings. Again, these tropical trees quite often produce false rings, what are also known as pinching rings. Those are rings that don't actually go entirely around the circumference of the cross-section. And these trees commonly produce rings that are just very difficult to cross-date with other trees in a given area. So the next few slides we're going to look at are some techniques that have been developed, very innovative techniques used to maybe overcome some of the challenges involved with tropical tree ring dendrochronology. This sample is known as Heidi commonly and C. hibiscus. I was able to sand this piece and again look at the heartwood and look at the rings, but dendrochronologists are using much more sophisticated methods than this to get to know more about temperature signals in tree ring chronologies. One way involves looking at lignin, and remember, lignin is what basically makes wood hard. It's a density. It's a substance that increases density in wood. So, using X-ray based blue intensity density research, this allows researchers to measure the amount of lignin that exists within the late wood. So researchers are shining light from the ultraviolet spectrum, and the amount of blue light that is reflected determines how much lignin is in the tree, and therefore how dense those rings are in the tree. So wood cells that reflect higher amounts of blue are less dense and contain less lignin, and those that reflect less blue light contain more lignin and are more dense. And this has to do with evapotranspiration, which we'll get into in a minute. Tropical tree dendrochronology may be at the frontier of the field of dendrochronology. The authors emphasize that this procedure using blue light intensity may be the first of its kind. specifically the first of its kind, a study like this being done with tropical trees. But it has produced evidence that tropical trees may be cross-dated using this blue intensity of the light spectrum. This research using blue light intensity, looking at lignin concentrations, is really tying in a lot with temperature and evapotranspiration. So tree physiology in the tropical environments is really at the mercy of extreme heat and evapotranspiration. In tropical environments, heat is the primary challenge for lumen cells. And as temperatures increase, so does evapotranspiration. So the authors explain that tropical trees must guard against what's called cellular cavitation. And that would be similar to an embolism. So if you imagine water being brought up from the roots, going up the tree through the xylem, up into and out of the leaves. Well, if there's not enough water supplying those roots, then the pull continues and you could get a gap in that water cohesion and water tension. And at that point, you would have cavitation or an embolism in the tree. And this will basically kill the lumen cells. And so the cells apparently increase the amount of lignin in those lumen cells, and in effect, they seem to squeeze those cells down a little bit, making them more narrow and reducing the possible impact that increased evapotranspiration would have uh, on the process of moving the water through the tree. So this process of measuring lignin's density is able to extrapolate an association, a correlation between temperatures and a higher evapotranspiration and the density of lignin in these trees. In my literature review, I found that tropical tree dendrochronology does seem to be at the leading edge of the field of dendrochronology. Another innovative approach applied Seabaum peak radioactive carbon dating to a study that was published in the Journal of Ecology and Evolution. The authors explain that prior studies demonstrated that Seabaum peak dating has been known to produce verifiable tree ring dating within a range of about 60 years, and several species have been dated with this approach. Seabaum dating is a process that correlates wood samples with military bomb testing material that remains in the atmosphere since the 1950s. This approach again borrows from the field of mass spectrometry, and this method was applied again to a tropical tree ring research study published in the Journal of Ecology and Evolution. Additionally, this study also employed a statistical method known as bootstrapping that is sometimes used when there's limited data available, and a situation like that would be with tropical tree rings. This has just been a brief overview of some of the material that I looked at in my literature review. Some additional studies that I read about included correlating tropical cyclones with Spanish shipwrecks that occurred during tropical cyclone storms, and then correlating those storms with tree rings and damaged trees located in the area, and at the same time, those ships were sunk. So there is a wide variety of research going on in this field. The first international tropical tree growth rate conference was held in 1980, and the field is continually growing as we learn more about tropical tree ring and tropical dendrochronology.